next speaker is Isabel Bustos, who is going to talk about beyond neurons, early steps towards becoming a neuroscientist. When you want.
Okay, so good morning, everyone. I am Isabel, and as my colleagues did before me, I'm gonna talk to you about a bit about my past and why or how I ended up here. But honestly, when I was a child, I didn't know that I would become a neuroscientist, or at least that I would try. Because honestly, I don't know about you, but I didn't even know that what a PhD was. So I just think that you know your path while you are doing it. So I just ended up here because of the last choices that I did. So everything started in high school when I become like really interested in biology, but more on a clinical part. But I didn't want to be a doctor because I was scared of blood and stuff. So that's maybe why I ended up here. So first I did a biotechnology degree in the Polytechnic University of Valencia. I didn't move along Argentina, but along the Valencian community. And then I had the opportunity to study abroad for 1 year in the Royal Institute of Technology in Stockholm, which was super cold, but super cool as well. And then I came back to do the first steps in a research lab, and I participated in a joint project between two institutes, the Principio Felipe Center and the Institute of Sanitary Research in the La Fe Hospital in Valencia. And then I moved more to the Valencian community, and I ended up here doing the Master in Neurosciences here at the Institute, where I stayed to do the PhD. So the first experience that I had in a lab, and more specifically in a neuroscience lab, was, as I told you, in Valencia. And in this case, I studied ataxia, which is a set of neurodegenerative disorders that mostly affect the cerebellum, and that have major implications because motor neurons are affected, and this has a wide range of clinical manifestations. The problem with ataxia is that there are many different pathologies and many different clinical manifestations, so the diagnosis, specifically the genetic diagnosis, is very complicated. And as you can see, there are, the different pathologies are not equally distributed around a population, so this even hardens more the diagnosis. So as I told you, the main characteristic is that the motor neurons are affected, and this gives rise to different symptoms such as gait imbalance and fine motor movements. So people have problems while walking and stuff. And there are other symptoms such as tremors, dysarthria, dysphagia, which are problems while swallowing and talking, or even nystagmus, which is problems where controlling your eye movements. So basically, the main objective of this project was to genetically diagnose these patients to try to establish a therapy as personalized as possible. So in order to do so, we were working with whole exome sequencing in TRIO to try to identify the genetic modifications that each of the patients had. And in some cases we were successful, and we were able to work with the doctors and clinically diagnose some patients, and in others we were not, but that's how science works. And then I came into the Institute of Neuroscience, and I moved into the field of neuroepigenetics, which I was not very familiar about, and I joined Ángel Barco's lab, the Transcriptional Epigenomic Mechanism of Neural Plasticity Group. And first, because some of you may be familiar, but others might not, what is epigenetics or what is its relevance? So this is like the canonical image to kind of explain or show what epigenetics is, which is the one-tone landscape. Basically, in a pluripotent cell has like all the genes in a cell, and that same cell gives rise to many different cell types in the process that is called differentiation, that you all know about this. So in this case, epigenetics is the mechanism that enables to silence or some genes while expressing others, like in a time and spatial controlled manner, to eventually give rise to different cell types with the same genes, like all of them bear the same genes in the neurons. However, this mechanism obviously is very important throughout development, but also it is relevant, and it happens on a daily basis in the adult state, and it is relevant for processes such as learning and memory, and this is basically where the group is basically focusing. So, but part of epigenetics is a very broad field, and there are many modifications that you can influence the expression of the genes. But the first project that I was involved into was to study the chromatin architecture and its relevance in gene regulation. So obviously we are working with neurons, which are postmitotic cells, and they are not gonadified, so the genes are already located within the nucleus, and they are not going to move a lot from the position where they are, and obviously they are located, and some of them are in the periphery, and some of them are in the nucleoplasm. So there are like different compartments, which is like the so-called euchromatin or heterochromatin, and the euchromatin, for example, is more less condensed, so this enables the gene expression, or genes are more likely to be expressed, while in the heterochromatin it happens the other way around. So in this project we were assessing how the location of the genes influence gene expression in this case. And then for my PhD I moved into another project which is more working with mice and behavior, which was kind of exciting at the beginning. And I mean, it just really is very exciting. Very exciting. And I work with autism spectrum disorders, as many of you know, and they are a set of neurodevelopmental disorders, and the basic clinical manifestations that I'm sure you already heard about is altered language, repetitive or stereotype behaviors, impaired sensory stimulus, or impaired social interaction, which is the basic hallmark. And the problem with this set of disorders is that the etiology is not very well defined, and there are a lot of both genetic factors and environmental factors that have been associated with these disorders. So basically, our aim, or what I would like to achieve, is to kind of disentangle and establish a common mechanism between these genetic and environmental factors. So for that, we are using two different mouse models. One that explores the genetic factors, which is a CEP heterozygous model. CEP is a histone acetyltransferase enzyme, so deficits in this enzyme will lead to hypacetylation. And another model that studies environmental factors, which is injections with PPI, that it's a HDAC inhibitor, so this will lead to a hyperacetylation state. So with these two models, we can assess the role of epigenetics in autism spectrum disorders. So basically, we hope that the common link between this is epigenetics, and we are studying this. And that was everything. I just wanted to thank my lab for being so supportive throughout this journey. Also Silvia and Sandra for organizing this because as I mentioned before, I didn't know that I wanted to be a neuroscientist. I didn't understand neuroscience when I was little, and right now I am trying to. But I think that it's important to have like role models along the way, so you can just discover your path as you are going. So thank you so much, and thank you for listening. So you have any questions?
Thank you very much, Isabel. Questions or comments? Or Thank you so much, Isabel, for sharing your experiences and your sincerity. I guess that you are already experiencing some frustrations with the experiments, but that's good, you know, still, I mean, I think there's so much that you will encounter in the future, and all of these experiences, they will prepare you for that. So I think, I mean, what you say at the end, that it was like, you think it is important to have a little bit more of references. Not really, I guess nobody's going to tell you what are you going to be in the future or anything like that, but at least that you have like these reference figures. And I think this is something that it has been trying to be promoted quite a lot. I don't know if from your point of view as a student that's starting, you feel that you have more information about these reference figures. So if we are still stuck in Marie Curie, and that's it, or you feel that you're seeing more of these like everyday scientists in, I don't know, in the media or in your, I mean, or not, perhaps not friends that are scientists, but you know, like they know and they can recognize Spanish scientists that have been made much more than before, or we are not doing a very good job.
I would say that there's a difference in the involvement that you have. I mean, for general society, maybe it is more common to know more male scientists, and the general public, if you ask them maybe name me a few renowned scientists that you know, probably most of them are going to be males, but I think that as you are more involved into science, obviously there are great male and female scientists, and you get more like examples. But I think that the more limitation, well, as I see it, is more the general public, because now obviously all the seminars that we have, when you go to another lab or whatever, you see that there are a lot of great researchers, both males and females, but I guess that it's more like the general public's view that is lacking the examples, I would say.
Okay, thank you.
I have a question, very general question about your thesis project, a couple of them. So first of all, when you showed briefly the risk factors, there is one that I don't know, there is less than 12 months between pregnancy.
Yeah.
So meaning that a brother that is born with less than, it seems like, I don't know, I'm not in the field, so it was quite unexpected. It looked like something a bit random, you know?
Yeah, I mean, I'm not a doctor, so I'm not sure about like the exact effect that might have, but yeah, there's like correlations between the shortest interval between pregnancies and autism. Maybe it's time recovery of the body. I'm not sure. I mean, that's more like a clinical perspective that I'm not sure about. But yeah, I was surprised too when I read that.
And then the other comment is very general, and it's in a way, every time that you have an animal model, it's recapitulating, of course, only some aspects of the disease, because the human disease is something else. But without being in the field, without respect to the disorder, I guess this is even more, because even the pathology and the pathology, the spectrum, so it's so different between one child, one person, and the other. So it's even more, no? Do you think this is something that you need to justify, the fact that the animal model inevitably recapitulates only a subset of this subpopulation, or it's accepted in the field because this is how the field is?
I mean, totally, it is totally what you say, that obviously, as it is a spectrum, and mostly autism is associated with many other syndromes, so there are like many different clinical manifestations, but like it is quite established that the hallmarks are deficits in sociability and kind of repetitive behavior. So although there is like a lot of heterogeneity in the clinical manifestations, there are like some phenotypic traits that are kind of common between autistic patients. So that's like the main trait that we are recapitulating. Because obviously there are many different phenotypic traits. Yeah.
And then there's comments that the clinical manifestation, the fourth one, I don't know also the impaired sensory input.
Yeah.
I know like the other three are what you normally think about when you think about some spectrum disorder. In which sense, what very specifically is impaired?
It's more like the processing. I mean, autistic patients are usually like really sensitive to being, for example, loud noises or bright lights, and they are kind of more affected by strong stimuli. So that's basically it.
If there are no other questions, I think we can first of all thank the speakers, the first speaker of today. Thank you very much.